All right. Hello, everyone. My name is Lance Ramsey, and I'm the marketing manager here at Formulatrix. First, I'd like to thank everyone for taking the time to join us today. So welcome to session 2 of our protein crystallization automation webinar series. Here is a brief overview of our agenda for this session. We'll start out with a presentation from our application scientist, Dr. Mayank Aggarwal. Then we'll be hearing from Dr. Sagar Sridhara from Florida State University and Mr. Harrison Jones from the University of Texas at Austin, followed up by Dr. Sarah Bowman from the Hauptman-Woodward Institute and Dr. Janet Newman from the Commonwealth Scientific and Industrial Research Organization in Australia. At any time during the presentations, please feel free to enter your questions into the GoToWebinar interface, and time providing, we'll answer those questions at the end of the session today. All right, with that said, I'll hand things over to our first speaker, Dr. Mayank Aggarwal. Let me just give you the controls, sir.
Thanks, Lance. Good morning, everyone from Boston. This is Mayank. I'm sure I've talked to most of you guys, so you already know me, but if not, I'm the product manager and application scientist of protein crystallization department in Formulatrix. What I'm going to talk about today is how, as a company, we have progressed since our inception and what advancements we have made in the last 3 years, plus how do we see the future of RockMaker. And again, I'm sure most of you have heard of and are using RockMaker software. So let's get moving. Let me start with a quick timeline. The company was started it was founded in 2002, and the first product we came up with was RockMaker software. And since then, in the last 16 years, we have come up with many imagers. We have come up with the formulator, screen builder, NTA drop setter, Sonic, which is used for micro crystal detection. It's a very sensitive and very advanced instrument. Then FRAP, which is used for LCP pre-screening, very useful for membrane protein crystallization. And then in 2016, we came up with, we integrated multifluorescence imaging into our imaging systems. And this year, we came up with some major updates to our Sonic, and we are calling it the Supersonic. And the newest product in the market is Pulse. It's not out yet, but I'm going to talk about that a bit as well. So the three things I'm going to focus on is MFI, Pulse, and Supersonic. And what is MFI, or multifluorescence imaging? Most of us, we use UV imaging to distinguish between protein crystals and salt crystals, but UV imaging is entirely dependent on the amount of tryptophans you have in your protein, right? So it's not uncommon to have a protein that you're working with which doesn't have tryptophans or which doesn't have enough tryptophans to give you a good enough contrast. So what you can do then is label your protein with a fluorescing dye, and there are multiple dyes available in the market: fluorescein, Texas Red, Alexa Fluor. So you can choose from a variety of commercially available dyes. So you label your protein with a dye, and that enhances the signal detection multifold. As you can see in this particular example, there are crystals which are hidden underneath the precipitate, but it's not visible under the visible light path. But because the protein was labeled, when you image the same drop using fluorescein fluorescence, you can clearly see those crystals. The other important application of multifluorescence imaging is if you are studying protein complexes. So let's say you have protein A and protein B, you bind them together, set up a crystal plate, you get crystals, but you're not sure, you can actually never be sure that the crystal you have is a complex crystal or is it a crystal of protein A or protein B until you collect the data and solve the structure. So what you can do then is, bind protein A with dye 1, bind protein B with dye 2, and then mix them together, and then let them sit for 5 minutes and give them time to bind to each other, and then set up the crystal plate. Now when you get a crystal, and if the crystal shows up in both the wavelengths, you are certain that the crystal is a complex and not just a crystal of one single protein. The second part of the presentation is about Sonic. As I've said before, it is an advanced instrument that is highly sensitive to detection of microcrystals. And Sarah Bowman is going to talk about this Sonic thing as well in the end of the, I think she's the third presenter after me. This is a well-known example by Formulatrix. We have been using it for many years now for advertisements. As you can see here, in the sample it looks predominantly clear with a little bit of light precipitate, so under visible light path, so you don't really know if there are crystals. But when you image the same drop using Sonic, you can see there are many microcrystals hidden. But when you dilute the sample 200 times, you don't see those crystals anymore because the signal is just too weak. So we have enhanced the the sensitivity of sonic 5 times, and as I said before, we are calling it supersonic, the signal detection has gone up multifold, well, fivefold actually. So now this can benefit you in two ways. You can either increase the sensitivity by keeping the exposure time the same, or if you are a really high throughput lab and exposure time is critical to you, then you can reduce this, you can reduce the exposure time but keep the sensitivity the same. So to each his own. Rockmaker. Now we are moving towards an era where instruments and hardware is not the only thing that governs or dictates your automation. We are moving towards a software-controlled automation systems, right? And Rockmaker is a very powerful, easy-to-use software that has a complete control over your whole crystallization workflow. You can go from screening your experiments to imaging your plates, scoring them, analyzing them, optimizing potential hits, and then repeating the whole process until you finally get a crystal. Once you do, you can print out reports, various kinds of reports, and share them with your colleagues, collaborators, your bosses, and the scientific community. Every year we try to release at least 2 versions of Rockmaker with newer features, with bug fixes. And the next version, which is coming up, would be in January 2019. The 2 major updates that we have in this feature are crystal tracking and machine scoring. Crystal tracking is a feature that would enable you to document your crystals for a synchrotron trip. So what it means is you can you can assign a crystal to a certain puck, to a certain doer, to a certain pin position within a puck when it is going on a trip, and you can always keep a track of where the crystal is, whether or not the structure was solved, what's the resolution, and where is the structure deposited. So I'm going to show you a few mock-ups of what we have so far. Again, this feature is still, actually, it is implemented in Rockmaker Enterprise Edition, but it is not completely implemented. So what I'm going to show you may or may not be in the version that you are using. Okay, so the first thing you have to do, and this is something you'll have to do only one time when you start using the feature. You have to define all the pucks and all the doers that you have in your lab. Of course, if you buy a new puck or a new doer, you have to define it again. But every puck and every doer has to be defined with a unique barcode and a unique name. And then you have to define your synchrotron trip. So let's say you are going on you have a trip coming up in the next 2 months. You define that trip, and then you start navigating through your crystals, through your plates. As you come across a crystal, you would click on this Harvest Crystal button and draw a box around a crystal, and that would open up this window where you would fill in more information. So you'll assign this particular crystal to a trip, to a doer, to a puck, and then to a position within the puck. And all these fields are linked to each other. So Rockmaker would know that puck number 1 is a part of doer 1, and doer 1 is going to first trip. You can also enter more information if you like. It's not mandatory, but if you are meticulous with your notes, you can add information about cryoprotectant, about soaking, and I'll get to these fields later. But then the workflow is like this. Once you have assigned enough number of crystals to a trip, then you would print out a trip report. And this trip report can be printed in an Excel file, as a CSV file, or as a cleaner PDF file. But you'll print it out as an Excel file first, and then you'll go to your lab and actually start harvesting crystals. So you'll harvest crystal number 1, which is from a certain plate, a certain well, and all that information will be a part of crystal ID. And then you'll freeze that in pin number 1 of puck number 1, which will go into doer number 1. Now, when you are doing that, of course, mistakes are bound to happen. So let's say crystal number 3 breaks, and now your numbering here on would change, right? So crystal 4 is not really a part of pin 4, it's a part of pin 3. So nothing to worry there. All you have to do is make that note in your printed sheet, and then when you have time, go back to the computer and fare up those changes in this CSV file on the computer. Once you are done, you will upload this file back into Rockmaker, and Rockmaker will update all the assignments of those crystals that you had harvested. Now, once you are ready to ship your crystals, you would send the printout of this particular CSV file to your synchrotron trip. And while you are collecting data, you'll fill out the column F, G, H as well. So you'll fill out the screening date, collection date, and the resolution of the structure. And once you deposit the structure, you would just fill it up with a link, with a hyperlink to that depository. It could be PDB or it could be your local storage area. And then you have to upload this file back into RockMaker once again. So when you do that, all these fields will be updated. So the screen date, collection date, resolution, structure, URL, and quality of the crystal will all be updated. Of course, you can also add attachments like SCAR files, your log files, and MTZ files into this one single window. So what we are trying to do here is evolve RockMaker into a more electronic bookkeeping system. And the next feature that I was going to talk about was Marco. It's an auto-scoring algorithm. It was developed by an employee of Google. So Marco is an algorithm that classifies your drops, your images, into each of these 4 categories. So actually not each, but it tells you what's the probability of a drop being clear, precipitate, crystal, or if it cannot classify it in any of these categories, it classifies it as other. And Janet Newman is going to talk about this in a bit more detail. Now coming on to the final slide, this is our newest product in the market. It's called Pulse. It's based on tangential flow filtration system. It's a very small instrument with a tiny footprint, and the utility, the function of this product is to concentrate your protein and buffer exchange it right before you are setting up your crystallization experiments. It's a true walkaway solution. So most people, what they use is a centrifuge, right, when you're concentrating your protein, which uses a dead-end filtration concept, and which works fine, except that it involves a lot of human intervention. So with this, all you have to do is load your samples in here in Falcon tubes, and this is the membrane chip that goes on the top of it. This membrane has different cutoffs, so you can choose whichever you like as per your protein size, and then program your cycle in the on the Raspberry Pi system, and then walk away. And once your protein is concentrated, you'll know because you can track the progress on your smartphone. Now I'm going to show you a quick video of this whole of this instrument. So it's not to scale, so but it's a very tiny footprint. So here's how you would load your 2 Falcon tubes. We can take from 50 mil to 100 mil protein sample. That's where the membrane goes on the top of the Falcon tubes, and this is the LED screen which you will use to customize to design a protocol basically. And then and this is the way how it works. So it aspirates the protein sample from the Falcon tube, and the protein flows through the membrane. And while it's flowing through the membrane in a tangential manner, the protein gets filtrated and it gets concentrated. Now you can either do a concentration or you could do a buffer exchange, up to you. And then the concentrated protein is dispensed back into the Falcon tube. So the protein is constantly being aspirated and dispensed back into the same Falcon tube and constantly being concentrated. The remaining solution which is not needed is thrown away or discarded into a reservoir which is on the right side in this panel, and you can take this off and throw it away if you don't need it. So with that, I would like to end my talk, and if we have time for any questions, I would take it up. Otherwise, we can always email you back the answers to the questions that you asked. Thank you, Lance. Over to you.
All right, thanks, Mayank. Thanks for that overview of our automation solutions and a glimpse of what's new to come. So next we have Dr. Sridhara from Florida State University. I'm going to pass over controls here.
Hello everyone, my name is Sagar Sridhara and I'm a postdoc in Institute of Molecular Biophysics in Florida State University. The title of my short talk is Structural and Functional Basis of Biomolecular Assemblies. I would like to give you a brief overview of the projects that I'm currently working on and also the projects that I have previously worked on. So currently I'm working on one of the types of CRISPR-Cas systems called the type 3 CRISPR-Cas system, wherein it's a characteristic feature of bacterial immunity. The bacteriophages are viruses that invade the bacterial cell and utilize the host transcription translation system to propagate itself. The end result of the phage infection is the death of the bacterial cell. However, bacteria has its own defense mechanism through which it defends itself from the phage infection. That is achieved through CRISPR-Cas ribonucleoprotein complexes that specifically bind to the phage RNA transcript and acts as an RNase to degrade the viral RNA. To achieve this, the host genome encodes a set of Cas genes and CRISPR-Cas and a CRISPR array. And this CRISPR array basically contains several short spacer sequences that are arranged in between repeat sequences. It is these unique spacer sequences that are key to identify the invading RNA transcript. So in the event of the injection of the viral DNA into the bacterial cell, the host CRISPR-Cas RNP, or the ribonucleoprotein complex expressed from its genome, would identify and bind to the CRISPR RNA present within the effector complex. The binding event would trigger a cascade of events that would cause certain conformational changes in the ribonucleoprotein biomolecular complex that would activate its RNase function to degrade the viral RNA. So this is what I'm showing over in the second part of the slide. Moreover, this effector complex would bind to another unique protein to get activated as a DNase and targets the viral DNA. Thus, the ribonucleoprotein would have successfully degraded the viral RNA transcript and the viral DNA, leading to the survival of the bacteria. So this is just an overview of the CRISPR-Cas system. The aims of my current project is to structurally elucidate the type 3 CRISPR-Cas complex intermediates, wherein we determine the structural basis of events that lead to viral nucleic acid degradation. We also aim to understand the cascade of transition events of the type 3 CRISPR-Cas complex. Specifically, we want to understand as to how the ribonucleoprotein switches from an inactive state to an active state to function as an RNase, and also understand how this ribonucleoprotein could function as a DNase. To achieve this goal, we use a combination of biochemical enzymatic assays and structural biology tools, mainly the cryo-EM and of course crystallization. In this slide, I'm showing one of the chromatogram, size exclusion chromatogram of the formation of the CRISPR-Cas complex in vitro. Specifically, the type 3 CRISPR-Cas system is composed of 5 protein subunits and 1 CRISPR RNA, which acts as an effector complex of unknown stoichiometry of individual subunits. Here, the individual subunits and the CRISPR RNA are expressed and purified separately. And as you can see, the complex is formed on a size exclusion column. The chromatogram basically separates out several unique species of protein-protein complexes and also protein-RNA complexes, which are all of significant interest to us. We would subject some of these species that are above, say, 150 kilodaltons to cryo-EM, and while several subcomplexes, which are basically coming in the later part of the chromatogram, are subjected to crystallization experiments. If we are able to obtain crystal structures of these subcomplexes, they will, for example, facilitate the model building of electron density maps generated by the cryo-EM of the full complex. So, here I would like to end the current project overview over here, and I will proceed to the next slide, wherein I'm talking about my the project that I've previously worked on. So this is completely different from what I've just spoken. Here I would like to just highlight one of my previous projects that I was involved as a PhD student in Karolinska Institute in Stockholm. The aim of this project was to understand the structural and functional basis of human mitochondrial tRNA processing. So in this slide, you can see a schematic representation of transcription within the human mitochondria. The human mitochondrial matrix contains several copies of mitochondrial DNA, and this mitochondrial DNA is short, circular, double-stranded molecule of around 16.5 kilobases. The mitochondrial DNA encodes information of 13 essential protein subunits involved in oxidative phosphorylation, along with 2 ribosomal RNA elements and 22 transfer RNA elements. And the transfer RNA elements punctuate most of the mRNA and rRNA elements. The transcription of the mitochondrial DNA is performed by mitochondrial RNA polymerase, which is abbreviated here as POLRMT. So that would generate long polycystonic precursor RNA sequences. So basically, the transcription of 2 strands of mitochondrial DNA would give rise to 2 polycystonic precursor RNAs, and this scheme is basically showing one such polycystonic RNA. In this scheme, only one is shown, and the long RNA transcript must undergo 2 key processing events to liberate individual ribosomal RNA, individual transfer RNA, and individual mRNA elements. The 2 key events are basically the 5' tRNA processing by the mitochondrial RNA-P and the 3' tRNA processing by the mitochondrial RNA-C. So as the scheme pretty nicely shows here, 3 proteins called MRPP1, 2, and 3 are acting as a mitochondrial RNA-P, while the other 3 proteins, MRPP1, 2, and ELAC2, acts as a mitochondrial tRNase-Z. So the end result of these 2 events is basically that all of the individual RNA species can undergo their own set of maturation. And this is mediated by a lot of unique enzymes, and we are not going to go into details of each of them. But I would just like to highlight one such, one of the sub-projects that I was basically involved in, wherein we were trying to understand the crystal structure of MRPP3 protein, among many others. So we were trying to crystallize the full-length MRPP3 protein, but unfortunately we were not able to generate any crystals of the full-length MRPP3 protein. What we did was to generate a series of truncated variants of MRPP3 protein. So here you see a schematic representation of MRPP3 protein. It has a mitochondrial target sequence which helps the protein to make its way into the mitochondria. And it has 3 domains. The first domain is the N-terminal pentatricopeptide repeat containing domain. It has a central domain, and it is followed by a C-terminal metallonuclease domain. So when we did not get the crystals of the full-length MRPP3 protein, we generated several truncated variants by successive deletions of 1 or more PPR repeats. So one such variant, which started from residue number 207 until the residue number 583 crystallized, and we were able to basically screen all these different variants for crystallization. And finally, we got a very high resolution crystal structure for of the variant which started from residue 207. Thus, the optimization of crystallization and optimization of crystal hits have only positively contributed to my projects. And this is where I would like to end my talk. Thank you.
Next, we have Harrison Jones from the University of Texas at Austin.
Great. Well, thank you for having me. Hi, everyone. My name is Harrison Jones. I am an MD Ph.D. student in Jason McClellan's lab at the University of Texas at Austin. And today I'm going to be telling you about a project or a new crystallographic screening strategy we've been working on called Iterative Screen Optimization. All right, so quick little background regarding this here is the crystallographic phase diagram. So when you're setting up a crystal drop, your real goal is to identify protein and precipitant conditions that kind of hit this middle sweet spot where you can get nucleation and stable crystal growth. If your protein and precipitant concentrations are too high, you will be in this top right area where your protein will precipitate and crash out of solution. And if it's too low, you'll be in this bottom left area where your solution is undersaturated and you do not get any crystal growth. A common method for crystallographic screening is called Sparse-matrix screening, and this is where you can test 96 semi-random crystallization conditions, and you kind of pick a semi-random point on that crystallographic phase diagram for each condition and hope that one of them falls within that sweet spot, and you're able to identify a crystal hit. And typically, if you do not see a crystal hit, you disregard that condition and move on and test something else, even if you were very close and only just undersaturated or oversaturated. Another method that people tend to use is called free interface diffusion, and this is where you can set up your protein and mother liquor on opposite ends of a capillary, and over time these two will diffuse and mix, and this gives you a much finer slicing of that chemical space and a greater chance of identifying a good crystallization hit. However, practically speaking, when it comes to isolating individual crystals for diffraction experiments, this can be quite tricky. So we set out with a collaborative project with Formulatrix to try to take advantage of both of these methods. And the general strategy that we used is to set up a new crystallization experiment. You can then incubate that for some period of time. We chose 5 days, and then evaluate your drops. You can then assign a user-based qualitative score based upon that visual inspection. So you can assign a score of clear, representing an undersaturated solution. You can assign a score saying that we did get a crystal hit, or a score indicating that your protein has precipitated and crashed out of solution. And then the real key is to then modify the precipitant concentration of each of the 96 conditions based upon that assigned score. And then you can dispense a new optimized crystallization screen using an automated liquid handler, such as the Formulator 15, and then repeat these steps multiple times for multiple iterations to try to get the most optimized crystallization screen. So we designed a new 16-ingredient crystallization screen to be compatible with the Formulator 16. It included a couple different pegs, MPD and isopropanol, a variety of additives and salts, as well as a couple different buffers to represent various pHs. We then generated our own 96-well crystallization screen, which we termed Sweet 16 because of the 16 ingredients. And we tried to cover as large an area of chemical space as we could without being redundant through the iteration process. We also chose 6 different test proteins to see how well our method worked at generating increasing number of crystal hits. We chose 3 proteins that are relatively common in the crystallization field, including catalase, concavalin A, and lysozyme. And then we chose 3 proteins that were a bit more unique just to see how broadly applicable this method would be. And that included the respiratory syncytial virus fusion protein, which is a homotrimer, the modivizumab antibody fragment, which is a 50 kilodalton heterodimer, and then M-cherry, which is derived from bacterial expression. When it comes to assigning our qualitative scores, we separated this into 4 different scoring categories. The first would be a clear drop represented by this white circle, which just indicates that your drop is undersaturated. The second score would be a crystal represented by this red over here, indicating that you did get a successful crystal hit. The third is a light precipitate, which we represent with this light blue circle in the bottom left-hand corner. And then the fourth score is a heavy precipitation, which we represent with this dark blue circle. It's worth noting that because all of this is a user-generated scoring, there is some subjectivity, and there's some drops that can be difficult to call. So if you have something like phase separation, that can be a little bit difficult and vary from user to user, or something with perhaps intermediate precipitation, or if you have some sort of contamination within your drop. So these are some problems that can occur, although I'll say from our experience it wasn't too bad. One thing that's really nice about this iterative screen optimization is it's already incorporated into the RockMaker software. So when you are visually inspecting your drops within the RockMaker software, if you go to the scoring tab, there is a section of different scores for this ISO algorithm. So you can assign it a clear, crystal, fine precipitate, or heavy precipitate score. This here is the general workflow of how this ISO algorithm works. So here on the left would be, so this represents a single condition within that 96 condition tray. So for that single condition, we'll set up our initial screen, or our initial drop, which is indicated in gray here, allow it to incubate for 5 days, and then we would have a, my apologies, and then allow it to incubate for 5 days and then assign a score based upon visual inspection. So again, white would represent a undersaturated or a clear drop, red represents a crystal hit, light blue here would be a light precipitate, and dark blue is a heavy precipitate. And then based upon that score, for example here, let's say our initial score was clear, you can then optimize that specific chemical condition. So if our initial score was clear, so our second concentration, so C2, will be the original concentration that was in our first drop plus 10%. So really what we're saying, the algorithm here would say if the initial drop is a clear solution, then we'll increase the precipitant concentration by 10% and then redo the whole process. Similarly, if we use, if our first score is a light precipitate, we can then drop our subsequent precipitant concentration by 10%. Or if it's scored as a heavy precipitate, you could drop it by 20%. It's worth noting that within each of those specific percentage amounts can be customizable, so whatever is suitable for your own research needs. But the whole idea is the subsequent trays you'll be setting up are optimized based upon the scores of your previous trays. So a quick little summary of this is you'll have your 96 initial screening conditions. You'll dispense your initial crystallization tray and then evaluate your drops using Rock Imager and assign a user-generated score to all 96 of those conditions. You can then input this into the RockMaker ISO algorithm, and it will optimize each and every drop based upon those scores, and then you can dispense this plate in the formulator, set up your next crystallization screen, and then go through the whole process again where you can rescore that tray and then again optimize further. So a quick little summary of all this using one of our example proteins was we set up an initial screen here and got a handful of crystal hits, And then, as you can see, moving from left to right, during each iteration and subsequent optimization step, we are moving closer and closer, or we're getting a greater number of crystal hits. So again, the red squares here indicate successful crystallization conditions, and on the left, you can see we start with 6 crystal hits, and then during each iteration, we are able to dramatically increase the number of crystal hits that we identified. So our results for this, for our 3 different proteins, catalase, Con-A, and lysozyme, you can see as we move from left to right, so tray 1 through the iteration process to tray 4, within all 3 of these proteins, we saw an increasing number of successful crystallization conditions identified within our screen. It's worth noting that the y-axis is very different for each of these proteins, suggesting that some of them were more easy to crystallize than others, but the general trend held true that by using this ISO algorithm and iteratively optimizing our screens, we were able to identify increasing crystal hits for each protein. Similarly, if we look at this for the RC fusion protein, the motivizumab Fab, and M-Cherry, we see a similar trend where, again, during the iteration process, we were able to identify increasing number of conditions that gave us successful crystals. And then lastly, one thing that was particularly useful about this method is we, within each target protein, we identified multiple crystal morphologies. So I'm sure many people here have experienced the situation where you get a beautiful looking crystal and then you take it to the synchrotron and put it up for diffraction, and it just doesn't diffract at all. And one of the key strengths of this method is if you can identify multiple crystal morphologies, you have a greater chance that you will have different crystallization space groups or different crystal packing that may just lead to different conditions and have a higher chance that something diffracts to high resolution. You can get a good structure. So with that, a quick little summary is in many cases, the conventional sparse-matrix screens may sample chemical space too broadly. So you may be close to a crystallization condition, but if you're not right on it, you will skip that entirely. And then using computation and some modern liquid handling devices, we can now automate the process of generating some customized and high-throughput crystallization screens. And then we can iteratively optimize these crystallization screens on a drop-by-drop basis to help maximize the number of crystal hits that can be obtained from a given set of stock reagents. And with that, I'd just like to say thank you to everyone in my lab, especially Jason, Colin, and Daniel Rapp for the help with this work, all the people at Formulatrix, and then Peter Kwong at the VRC. And I'd be happy to take any questions now or at the end of the talk.
Okay. Thanks, Harrison, for sharing that approach to optimization. We'll save all the questions for the end if there's time. And so next we have from the Hauptman-Woodward Institute, Dr. Sarah Bowman.
Okay, thanks very much. Thanks very much for organizing this. I think it's been great. The morning session was great, and this afternoon's been great so far as well. So I'm going to tell you all a little bit about some of the things that we're doing at HWI in the High Throughput Crystallization Screening Center, and a lot of the work that we're excited about starting. So first of all, just as an introduction for anybody who is not familiar with us, we are in Buffalo, New York, and we are a center that is really available for anybody to use. So you can send your samples from anywhere in the world for us to set them up for very sparse screening. We actually set up in one tray instead of 96 conditions, 1,536 conditions. We use a microbatch under oil method, and this is just an example of one of the trays that we set up. And for microbatch under oil, what we do is we add oil to each well, and then 200 nanoliters of the chemical cocktail, 200 nanoliters of your protein, and then we spin those down to merge the drops under oil and have a robust imaging schedule over time to monitor the crystal growth. And so this is obviously a little cartoon of what that looks like. So one of the things that is a little bit different for us is that our crystals grow really only at the bottom of the plate in the merged drop that's under oil. And so we have to image our plates upside down. And so we turn the plates over, and our imaging technologies to actually detect crystals, we use both imaging tables for visible images that have been made here at HWI, and we also use a Rock Imager 1000 with Sonic. This is really our workhorse for detecting crystals and especially detecting nanocrystals, which we'll talk about as time goes on here. And Formulatrix was great in actually setting up adapter trays for us to be able to put our trays in upside down, as well as be able to image the 1536, because that's a little bit unique relative to a lot of the imaging that occurs in other labs. And for us, what we really want to be able to do is to provide these crystallization services for people who may not have access to some of these robotics and automated techniques, as well as imaging methods. And so again, at the end, I'll share our website information. So one of the things that is interesting about doing such large wells, such large plates, so many different conditions, 1,536 conditions that are imaged 7 times using the visible images, once using UV TPF and SHG, which we'll talk about in a second, is that for each sample that we set up, we actually generate a minimum of 13,824 images. And so that's a lot of different images to actually go through. And you'll hear a little bit about how we use the different methods here to really pull out the things that are generating crystals. So just really briefly, what does Sonic get you? Sonic in our Rock Imager 1000 has two additional features beyond visible imaging, and one is the UV ultraviolet 2-photon excited fluorescence, which allows us, just like any UV imaging, to detect concentrated protein. So that will detect protein crystals, as well as proteins that are in gelatinous states, precipitated, but it's dependent on the protein actually having tryptophans or aromatic residues. And we'll see some examples of this in just a second. Sonic also gives us second harmonic generation, which is something that will identify chiral crystals. And so these 2 photons, which are from the same laser source as the UV TPF, generate a second harmonic generation signal. Positive signals here are high contrast and white against a black background. They will detect most space groups. And so what's really great about this is that they can detect submicron crystals or nanocrystals, which are very helpful for things like X-ray free electron lasers. So again, what we have is, oops, I went a little bit too far. What we have is our 1,536 conditions. We image these visible UV TPF and SHG, and how can we use these multimodal images to actually detect protein crystals? So first of all, it makes identification of protein crystals relatively straightforward. And what you're going to see in the next number of slides are a series of images where we have visible UV TPF and SHG images. In this case, it's actually quite clear that we have protein crystals in all 3 of our modalities. We can also use it in images of our wells that may not be as clear that these are protein crystals. Again, in this case, we can clearly identify that these are protein and that they are crystalline. Not all crystals will give a positive SHG signal, so there are some space groups that won't light up in SHG, but again, we can see visible and UV TPF. And even sometimes when the crystals may be below precipitate, we can see the bright spots. And in this case, we can actually rule conditions out. So this actually helps us a lot. Negative conditions are helpful. And we can also identify probable salt crystals. So in this case, we have these lovely little crystals that we wouldn't want to optimize because though they are definitely crystalline, they have no UV TPEF signal. So we can also detect nanocrystals. So this is, again, really exciting for anybody who is doing work with X-ray free electron lasers where we need very small, homogeneously sized crystals and a lot of them. So in this case, you can just barely see the crystals here, but using the UV TPEF and SHG, we can identify these crystals. This is one of my favorite examples. This is one of the only actual well in this 1536 tray that generated any crystal hits. You would not necessarily identify this as crystalline, but using the UV TPEF and SHG, we can see that these are actually beautiful nanocrystals that are growing very nicely. So I want to just end the last few minutes with some new advances. So one of the things that I know that Mayank already talked about was that there is a new photomultiplier detector for enhancing the SHG signal. This is, again, really helpful for detecting nanocrystals. This is an example of one of the wells from one of our 1536 trays that we actually sent out to Formulatrix where they tested for us how well it does. And really, we do see an enhancement of about 5 times for most of the wells that we might not otherwise identify crystals in. And I'm going to end by talking about some new work that we're doing in collaboration with scientists at SSRL and LCLS to actually do some room temperature sample mounting and actually do in situ crystallization to be able to develop crystallization that we can then ship to a synchrotron or to an XFEL source and able to set it up directly onto the beamline without any manipulation or handling. And so the goal here was to actually generate a robotic compatible crystallization plate. And it has actually already been implemented. You're going to see some prototypes in just a second. Essentially what we do is these wells, there's a number of different wells that we can set up, and each of these wells can contain a variety of different types of pins. This is one of the pins that they have developed at SLAC, and essentially you can set up, as you can see, hopefully a large number of different crystallization conditions. You can then image these wells and be able to know exactly where your crystals have grown, mount them directly onto the beamline and be able to collect data on these. And there's a variety of different kind of compatibilities. What's really cool about this is that we can actually ship these things across the country. So first of all, there was an initial setup for how the different how these might be shipped, and these were developed by some interns at SLAC, at SSRL, to be able to figure out how we could actually set this up to be able to ship room temperature crystals with really no manipulation. So this was the prototype, and actually we tested this about 2 weeks ago. This is now the prototype. So here's an actual image of the tray in the blue box here, and they're being set up in a dewar, mostly to keep it upright. So it really requires a Hampton-style magnetic base. If the sample base fits in this little tester that's shown in Figure B, then it will fit within the plate. And so in this situation, we can actually set up crystallization, a variety of different conditions. We can store these plates. We can have them humidity controlled. We essentially put the mother liquor in below this with some agar, so it doesn't, it's not actually liquid, but it keeps it humid. And in fact, we've shipped samples from California to here at HWI in upstate New York, and then ship them back. And actually the crystals last beautifully. They do wonderfully, and I think I'm out of time. So I know some of the other developments, I believe Janet's going to be talking about the Marco project. We're also doing some other things, and with that, I will say thank you for your attention. Our website is www.getacrystal.org, so you can look us up if you have further questions. Send me an email. Thank you.
All right. Thank you very much for that overview of what the High-Throughput Center has to offer and the results that you guys are getting. Next, our last speaker of the day is Dr. Janet Newman from the Commonwealth Scientific and Industrial Research Organization in Australia.
Awesome. So nobody says Commonwealth Scientific and Industrial Research Organization because it takes 10 minutes. Everybody calls it CSIRO. So,
CSIRO it is.
Yeah, I'm from CSIRO. It just makes it easier. All right. So what I wanted to talk about today was one of the collaborations that we've had to do machine learning and artificial intelligence to recognize the output of a crystal experiment. And I'm sure somewhere down here, I shall see next slide in the fullness of time. Yeah, okay. All right. No, yeah, here we go. All right. So the big thing is that when you look at something, you don't necessarily see what's there. You see what you think you see. And this is a problem that we have with crystals. And this is an age-old picture, whether you see a duck or whether you see a rabbit. Depends on what you're looking for, and that happens with crystallization experiments as well. So let's think about then what actually goes on in a crystallization lab or in a crystallography lab. What crystallographers want is they want crystals because they need to solve the structure. They're not actually very interested in, or they don't have the luxury of being interested in working how to get crystals more efficiently, because unless they get structures, they don't get funded. And so finding out better ways of getting crystals is a luxury they really can't afford. And that's where people like Formulatrix, people like the facilities are so important, because their focus is on improving crystallization for the benefit of everybody. All right. So one of the ways we could get better at crystallization is learning from the past. And the way we would do that is we would need to have an input for each experiment, what the protein was, how it was prepared, what its temperature was, all the parameters of the experiment. We need to have an outcome in terms of what happened when we set it up in a crystallization trial. And then, of course, we'd need to find correlations between the two. And that sounds sort of obvious, except and then once we had that information, we could start answering questions like, well, when should we start looking for crystals? And, is there a best temperature, and is there a better chemistry? And, how often do crystals grow and then dissolve, which they do, and it's depressing. How can we tell when something isn't going to work? When can we stop doing experiments? And, how do we optimize most efficiently? So what we need is data. What we have, input data would be the parameters of the experiment, and the output, what we need is some sort of description of what happened. What we actually have, though, at the moment are pictures. Well, that's fine, except we can't use them to develop these optimization techniques. Sometimes we don't even have crystals. I mean, we don't even have crystal pictures. We just have trays, and that's even worse. So if we're going to try to translate the outcome of a crystallization experiment into a scheme that can be used for data manipulation, there needs to be some sort of standards of how to do that. And if you look on the web for ways of interpreting a crystallization image, you will find some interesting information. None of it actually matches. So there's no consistent set of rules for interpreting a crystallization experiment. And when we look at all of the things which are available on the web, we find that they can all be collapsed down minimally to a set of 4 categories, and that's clear, precipitated, crystalline, or other. And as many of the speakers have already pointed out, that clear generally maps being undersaturated or indeed undernucleated. Precipitated means that you've got at least a saturated solution, or you might have a denatured solution, and a crystal, of course, is what we want. Other is the bin where we put everything which we don't quite understand how to classify into those first 3 categories. And that's just what I said, that, if you have just those 3 categories of clear, precipitated, and crystal, then you can more or less map out a phase diagram. So we've tried to use machine learning to classify the images that we generate in our center at CSIRO. And we've tried a number of different things. The first thing we did is we thought our users will be interested in looking at their crystals and scoring, and we found out that that was just not true. So of all the crystal images that we've collected, and we've collected about 65 million of them, only about less than 2% have actually been given a user score. Then we tried some of the existing scoring strategies which are out there. So Bezra came from Hauptman-Woodward, Alice came from University of York, TextRank came from Oxford, and we found that all of these required a lot of training, and of course we don't have many examples for training because our users don't score. So what we did is we developed an app. We call it Cinder for Crystallographic Tinder. And the idea of that app is that it will just present images to anybody who cares to open this app, and you can score them, and we would get images for a training set for machine learning. And then we would use all the information that we had, user scores, Cinder scores, anything that we could get, and we would develop our own scoring algorithms. And at the time we were doing this, it became obvious that deep learning was going to be the way to go. And so we started doing some in-house deep learning experiments, and then we discovered that there was a company in California that was specializing in this that was called Deep Crystal, and we worked with them, and we got some machine learning workflows up. Wasn't really very good, but it was okay. And then while we were doing this, Patrick Charbonneau of Duke University started bringing together a group of people and we formed the MARCO Consortium. And the MARCO is machine recognition of the crystallization outcomes. And in this consortium, we had people who have a lot of crystallization images which are scored. We had access to people from Google Brain, and we had Patrick who organized it all. And what we decided over the course of this collaboration is that we were going to stick to our 4 basic classes. We were going to use as many images as we could get that were scored, and it turned out we ended up with 500,000 scored images. These were expanded by random permutations to a set of about 100 million different images, which were then used in a deep learning algorithm. We used both vapor diffusion images and microbatch images, and the microbatch images, all 170,000 of them, were contributed by the HWI. And you can read about this on that link that I show here. It turns out that MARCO, with this amount of training, was very, very good. So we get over 90% success rate of correct scoring of these images using the MARCO algorithm. Now this compares to humans, who generally, it's been tested a couple of times, are only about 80% reliable in actually scoring outcomes. So now already our machine learning is better than most humans. Then we tested it independently with some images from CSIRO, which had never been anywhere near the MARCO algorithm. They weren't used for training, they weren't in the validation set. And we found out that the machine learning that we were using at the time, which was the Deep Crystal, was overall about 74% accurate. And then MARCO on the same set of images was 90% accurate. And this is amazing because many of the images that contributed to the MARCO algorithm weren't vapor diffusion at all, and we use vapor diffusion. So it turned out that the information from microbatch was enabling us to get much better results with vapor diffusion experiments. So how do we do this in our lab? We have an automated system set up where periodically every hour we scan our database for newly collected images. This is then put through a cron job and a bunch of different, just queues, I guess, using Slurm and TensorFlow onto our GPU cluster. And then a score is estimated and then passed back into the database in real time, essentially. So it means that we get all our scores back within an hour of everything being imaged. All right. So it turns out that MARCO is good. In fact, MARCO is very good, but MARCO isn't perfect. So what you see here are 2 examples of MARCO scoring, and we show the users the MARCO scoring by putting a pale colored band around the image. So if the pale color is a pink color, then that would suggest that MARCO has scored this as a crystal. On the right-hand side, you can see that there's a pale blue band around the image, and that suggests that Marco has scored it as precipitate. So the image on the left, which Marco says was crystal, is probably because there's a feature at around 9 o'clock on the image, and that's actually a feature from the plate. That's just an error in the molding. It's a molding defect on the plate, and it's not a crystal at all. That's actually a clear drop. On the right, we actually have crystalline precipitate. So what we're doing now is we're working on using inline training to take images which have been scored by Marco and scored by a human differently, and then using those to actually tweak Marco to get even better. And this is where I'm going to ask you to help. So our app Cinder can be downloaded for either iPhone or for Android. And what there's 3 different sections for Cinder. There's Cinder Solo, and that allows C3 users to look at their own images, and so that's not relevant unless you're a user of C3, which is our lab here. Cinder Community is a section where the images which are mismatches between human scores and Marco scores are presented to the community so that we can get consensus score for our inline training. And then finally, if you know people who'd be interested in doing this, but they claim that they don't know how to look at crystallization images, Cinder also has a training set of images which will teach you how to look at crystallization images, and this is called Cinder Kinder. And there's about 200 images on there. And so how does this all help us? Well, from this month, I looked at some of the numbers in our database. We have about 4 million drops that we've set up, and we have 65 million images, of which 47,000 are visible light images. We've had just over 500,000 manual scores, and that number of scores has been collected for the last 12 years. We've had 30,000 Cinder scores, and we've had well over 2 million Marco scores. So we're making a huge difference by having this machine scoring in our lab. Then finally, if you have both data, input and output data, you can start building tools which enable you to generate hypotheses. And this is one of the tools that we have to do this. This is our Visualize Phase Space tool, which is available on the web. And you can see here, we have a set of parallel axes, and the parallel axes define a crystallization condition. So you have a concentration, a unit, and a chemical. And each line through our parallel axis plot is a crystallization condition. These lines now have been colored by whether it's a clear, a precipitated outcome, which are in blue, or a crystal outcome, which is in red. And so looking at the data plotted in this way, I can see straight away that the thing that affects crystallization and gives me crystals rather than precipitate in this case is the pH. So as long as my pH is above 8, I get crystals. And if it's below 8, I don't. And that then allows me to start really getting effective in my crystallization. And then, you know, another thing that we've used our Marco scores for was we set up 2 subworld plates. And we've always been a little nervous about whether the time lapse between setting up the drop number 1 in a 2 subworld plate or the drop number 2 in a 2 subworld plate actually affects the results. And what I'm showing you here is the Marco scores of our test plates where we set up lysozyme. We set up lysozyme test plates at least once a week. And what I'm showing here is the scores, the days to the first crystal in both the top subwell and the bottom subwell. So you can actually see that there's a pattern of repeat. So every third column takes longer to crystallize, and that's okay because every third column has a different crystallization condition. What you also notice is that there's no difference in the time it takes to get crystals in the top subwell or the bottom subwell, which tells us actually that there's no difference between those two in the way that we're setting things up at the moment, so that we don't need to worry about that particular aspect of the time being important, which is a relief. And then finally, what I'd like to do is not only acknowledge the people in the Marco Consortium who have produced an amazing tool, but also the people at CSIRO who have built some of the tools that we use, and of course the people from Formulatrix who have given me the opportunity to talk about it.
Okay, thank you so much for your presentation and all your insights and how machine learning can really aid in crystallization. It looks like we've exceeded our time by a little bit. For those who have submitted any questions We will try to follow up via email to make sure we can get you those answers. All of the talks today have been recorded and will be made available on our website to watch. Finally, I would just like to thank everybody for joining us today, and a special thank you to all of the speakers. Excellent talks today, fantastic. And so with that, we'll close session 2. So have a great rest of your night, everyone.